A formulation team spent 14 months optimizing their microsphere formulation, refining drug loading, tuning the polymer ratio, hitting every internal specification. 3 months before their IND submission, they delivered their in vitro release profile bore almost no relationships to what was happening in vivo. The entire analytical strategy had to be rethought, not revised, rethought. That program lost nearly a year. We have seen that specific scenario more than once. Hello, I'm Raghuveer Singh Parmar, Director Scientific Affairs at Resolve Mass Laboratories, where we help development teams navigate these kind of exact challenges across every major platform in long-acting injectable development. Long-acting injectables are one of the most commercially consequential formats in the parenteral development and one of the most technically demanding as well. When you replace a daily oral dose with a single monthly injection, you are not just improving convenience, you are altering an adherence dynamics, plasma concentration predictability, and therapeutic outcomes. For conditions where missed doses carry real clinical consequences, psychiatric medications, antiretrovirals, and hormonal therapies, the long-acting injectable format changes the treatment paradigm. That creates pressure. When the stakes are this high, the formulation science has to match them. How controlled release works. Every long-acting injectable centers on one principle, the depot. You are engineering a reservoir inside the body that releases the drug at a predetermined rate. The early phase is governed by diffusion, the drug migrating from higher concentration in surrounding tissue. As that phase is managed, material breakdown takes over, extending the delivery window across weeks. What separates a well-characterized depot from a problematic one is whether your in vitro method actually predicts in vivo behavior or not. Often it doesn't, and not without a dedicated method development built specifically around your formulation for this particular problem. PLGA microspheres, the industry standard. The PLGA microsphere, polylactic and co-glycolic acid, remain the most established platform in this space. The polymer is biodegradable, the regulatory precedent is solid, and release kinetics can be tuned by adjusting molecular weight, copolymer ratio, and the encapsulation method. The drug is trapped inside porous polymer particles. Water enters, triggers hydrolysis, and polymer degrades on a timeline you define, weeks to months. That's the theory. In practice, three variables consistently challenge development teams: encapsulation efficiency, the initial burst release, and long-term stability under accelerated conditions. A team without deep platform experience can optimize one parameter and unknowingly compromise the other two. The interdependencies are precisely where development becomes genuinely difficult. We once worked with a team developing a peptide-loaded microsphere intended for a twice-yearly dosing regimen. The molecule was highly sensitive to moisture during encapsulation. Even fractional humidity exposure during spray drying was causing aggregation that was invisible in early characterization but appeared clearly in the 3-month accelerated stability data. The entire process had to be re-engineered under tightly controlled atmospheric conditions. It wasn't on anyone's risk register. For molecules with this secondary structure sensitivity, it rarely is, and until it becomes unavoidable. Now, liposomes, drug delivery through membranes. Liposomes operate in a different design space. A phospholipid bilayer, structurally analogous to the cell membrane, encapsulates the hydrophilic drug in the aqueous core or hydrophobic drug within the lipid cell. For long-acting injectable applications, pegylated liposomes offer extended circulation and controlled tissue distribution. The challenge is consistency. Particle size distribution, lamellarity, and encapsulation efficiency must meet tight specification across every batch. These aren't minor deviations, they are the difference between a product that demonstrates bioequivalence and the one that doesn't. In situ gels and implants. In situ forming systems take a different approach. You inject a liquid, the body's physiology, a temperature shift or solvent exchange trigger gelation or precipitation, forming a depot at the injection site. No prefabricated particles, no surgical implantation, and high drug loading is achievable. The analytical complexity, though, is real. Characterizing gelation kinetics, understanding how depot morphology varies across injection sites, demonstrating syringeability across a clinically relevant temperature range, all require purpose-built methods. Standard compendial approaches don't serve these systems. Crystalline suspensions and lipid carriers. For poorly soluble compounds, most current development pipelines, crystalline nanosuspensions offer a path where classical approaches fail. Particle size directly controls resolution rate and therefore duration of action. Lipid matrices work through a related mechanism but introduce additional complexity. Polymorphic transitions during storage and the effect of excipient purity on release behavior. These variables surface in stability studies, often at the worst possible point in the development timeline. Now let's talk about next generation hybrid systems. The field is moving toward hybrid platforms. PLGA cores with liposomal coatings, crystalline particles embedded in gel matrices, multi-component depot systems that combine the strength of individual technologies while introducing new analytical and regulatory challenges. FDA expectations for characterization of novel combination systems are still evolving, and the teams new to this territory are largely without a map. Three things consistently determine whether an long-acting injectable program reaches the submission with clean, defensible data. First, platform selection must match your molecule's physicochemical reality, not your team's existing infrastructure. Second, in vitro release methods must be developed to predict in vivo behavior, not merely generate data. Third, stability strategy must anticipate degradation pathways before they appear in formal studies, because finding them late is precisely what creates those year-long setbacks. Generate data. Third, stability strategy must anticipate degradation pathways before they appear in formal studies, because finding them late is precisely what creates those year-long setbacks. Long-acting injectable development rewards depth and punishes assumptions. The programs that reach submission with well-characterized packages are built by teams who invested early in understanding how their molecule actually behaves inside the carrier, not just on the analytical trace. If your program is at the stage where technology selection, release method development, or formulation characterization is creating uncertainty, that's exactly the kind of problem Resolve Mass Laboratories works through every day. Reach out to us if you are moving forward with this kind of programs, and we would be happy to support you in your program. Thank you so much for watching this session, and see you in the next one. Until then, goodbye for now.